A History of Biochemistry. Before the 19th century, chemists believed the world to be made of either living matter or non-living matter. They thought that the processes of life were unlike the processes of nature. They assumed life could not be explained by the laws of physics and chemistry alone. They argued that the molecules of life could only be made by the living organisms themselves. It was not until 1828, when the German chemist Friedrich Wöhler created an organic compound from inorganic matter, that the field of biochemistry began to develop. It became a new branch of science dealing with the chemistry of living systems. Since Wöhler's discovery, chemists have made significant advances in understanding the chemical structure and functioning of biological molecules. Friedrich Wöhler was a 28-year-old chemist and medical scholar. He accidentally produced the organic compound urea while experimenting with certain inorganic compounds. Urea is found in the urine of most animals. His findings demonstrated that a compound thought to be created only by animate beings could be artificially synthesized in a laboratory. They thus disproved the theory that an undefined life force was needed for the production of organic matter. 5 years after the publication of Wöhler's results, the French chemist Alcime Payen discovered the first known enzyme. Enzymes are proteins that speed up or slow down chemical reactions in the body. Payen's enzyme was called diastase. Diastase is a type of enzyme responsible for the breakdown of starches into maltose. Starches are complex carbohydrates found commonly in potatoes and rice. Maltose is a sugar. Payen found diastase in a malt solution. He created the solution using grain sugar. In 1897, Edward and Hans Buchner of Germany furthered the understanding of enzyme function. Their experiments used extracted enzyme-containing material from yeast cells. The brothers demonstrated that the inanimate material could ferment sugar. They showed that dead cellular compounds can perform the chemical reactions of living cells. By the turn of the century, chemists such as Emil Fischer had contributed effective methods of separating and identifying basic biological molecules. These molecules included various proteins, carbohydrates, and enzymes. The actual field of biochemistry was not recognized, however, until the early 1900s, when Carl Neuber formally coined the term. Studies in biochemistry continued into the 20th century. The goal was to provide chemical explanations for how cells carry out certain metabolic processes. It was discovered, for example, that the presence of specific sets of enzymes within a cell will facilitate specific chain reactions. It was also discovered that carbohydrates share similar molecular structures and that proteins are synthesized by the bonding of simpler molecules called amino acids. The next major breakthrough in biochemistry did not occur until 1943. That year, an American physician and medical researcher, Oswald Avery, found the culprit of hereditary traits. Deoxyribonucleic acid, more commonly known as DNA, was first isolated by Swiss biologist Friedrich Mescher during the latter decades of the 19th century. By the 20th century, the structural form of DNA was already being studied. Avery's famous 1940s experiment transferred traits of one type of bacteria onto another type. He did this by mixing dead cells of the first type with live cells of the second. His experiment provided the first confirmation of the link between DNA and inherited characteristics. A decade later, scientists James D. Watson and Francis Crick gave the first accurate account of DNA structure. Then in 1957, they presented the process of genetic encoding in DNA. Their work demonstrated that the specific information carried in DNA was responsible for the synthesis of specific proteins. These proteins then carried out specific cellular functions to complete the encoding process. Biochemistry continued to advance with the development of new technologies and techniques. These included chromatography, which is used in the separation of mixtures, and X-ray crystallography, used in the analysis of molecular structure. Although these innovations have led to the discovery and detailed understanding of many molecular structures and metabolic processes of living cells, biochemistry remains a vast subject of study that seeks to explain how chemical elements are incorporated into highly complex organic molecules. The field is still expanding. Researchers continue to uncover the specific functions of these elements in living systems. Their research supports the theory that life arises from chemical and physical processes.